To finish out, we're going to ask Dr. Kutter to come up. She's going to talk to us about phaging Dr. Kutter, how bacteriophages will change the way we interact with bacteria and their human hosts in a post-antibiotic world. This has been a passion of hers since 1963. Really has put on 20 biennial evergreen international phage meetings, and we welcome her to the podium to update us on phage.
Well, first of all, thank you very much for inviting me to come here. It's been an extreme interest of mine for a long time to think about diabetics, ulcers, and things related to that. And I've thought for a long time that that's a place where we should and could be able to test phage in important ways, and I'll talk a little bit about why. It's the end of a long day, and it's hard to know where to begin on talking about something so complex, so what I'm going to do is do a little bouncing back and forth between historical things and putting things in context and see if I can get how intrigued I can get you as to phage. First of all, I was asked to talk about phage in a post-antibiotic world. I don't think that we're ever going to have a totally post-antibiotic world, and certainly phage wouldn't be the solution there. It's got some really good things and some things it can do and other things it can't. But I think what we can do with it, particularly if we manage to finally move ahead and actually implement it in some of the rest of the world besides the Republic of Georgia, is to push off indefinitely a post-antibiotic world in many ways. And we need to look for those kinds of things and talk about an era of antibiotic resistance. A lot of happening, that interest is finally growing, and the need are growing. Time to move forward collaboratively in various ways. The news about things like superbugs that threaten a return to dark ages is on newspapers all around the world. Oh, hey, thank you. And that even nature is saying that phage therapy is getting revitalized. We have pushed towards a big phago burn study in Europe, which is for burn problems with Pseudomonas and E. coli, and that's something that's got millions of euro and 3 main countries and all sorts of things involved. There is work that Amplify is doing now with the U.S. Department of Defense, particularly with Walter Reed Hospital, to try to move things forward. But all of that is years before we'll get anywhere. And there must be a way to move things forward more quickly for certain kinds of things. And I think staph phage for diabetic ulcers is a very interesting candidate for a collaborative proof of principle trial. And just to give you a teaser now at the beginning, a couple of years ago I finally got reintroduced to a student of mine from 1975 who's Randy Fish, who's now a podiatrist, who was very interested in the possibility of using phage. And we have an agreement in a nonprofit Phage Biotics Research Foundation I have to be able to get phage from the Republic of Georgia that is commercially approved and sold in pharmacies there. And we can get it for the cost of shipping it for these kinds of trials. In Washington and Oregon, that's quite useful. We've been doing it mainly with naturopathic physicians for gastrointestinal problems, because there what you have is that they have the legal right to use natural products that have been extensively used in other parts of the world. And we're using that same principle in doing these preliminary diabetic ulcer kinds of things. And as you can see here, this was a patient who had something that had not been healing for over a month on courses of antibiotics, and very quickly healed when we started using phage. First of all, what's phage? They're viruses that kill bacteria, and what you have is a fancy icosahedral head that's packed full of DNA, and attached onto a neat injection device that can transfer it from one place to another. And what it does is simply will only attack to very specific kinds of bacteria. It injects the DNA into the cell, and then you have a series of things that happen, and exactly what depends on the phage, but these large lytic phage tend to make, first of all, a whole group of small molecules that I call monkey wrenches that are able each individually to turn off the bacterial host. And when we were trying to do the genome of T4, there was a big hunk that was hard to complete because we had at least 50 different genes, each of which individually were host lethal. So you have something which is both very specific and very lethal. And then what happens is that they go ahead and convert this factory that was making Cadillacs to a factory that's making motorcycles, and within half an hour you have hundreds of new bacteriophages released, which can then go deeper and find more cells of the target type. I first saw phage therapy happening when I went to the Soviet Union as part of finishing up the T4 genome project, and got to know the Eliava Institute and the areas where they've used phage a great deal since the 1930s. And it was there that my first actual introduction to seeing it happen was with a diabetic ulcer. And it was somebody who had a very bad infection and had come in from the mountains to have his foot amputated. And Guram Vasalia, the chief surgeon there, said, please let me try to treat it. Interestingly, you almost never see people who are amputated around Tbilisi. I think that, and you don't see a lot about diabetic ulcers there because they seem to be treated very early with the phage and not get much worse. So this is the treatment there was, what I saw was 3 days after they had done the first treatment, there wasn't a sign of purulence anymore there, and it went ahead to be cured. But what impressed me wasn't just that they were able to cure it, but that Vasalia was 95% sure that he could cure the infection part, and then with proper treatment of the patient, could help the patient to be fully functional. I saw a lot of kinds of, through the years that I've been going there, of use of it on quite extreme cases. This is one of the more extreme kinds of things, to see that it's something that's not just for the feet. This was a mine explosive injury, and most people who've seen this, most physicians say, why didn't they amputate? We'd amputate. And yet, within a couple of months, this man was able to use this arm, and the main difference between that and what we'd know was the use of phage, of a complex phage mixture to keep the infection from developing, and then being able to go ahead and get closure in a very short period of time. I had my first personal real involvement in it with a fellow who had come to one of our bacteriophage meetings in 2000. He had a very badly smashed ankle on a trip in Central America, and had wound up with a staph infection, and this was from 4 years earlier. And after 4 years, including a full year on antibiotic IV treatment, he still had staph coming out of both sides of his ankle. And the most interesting piece about this slide, this is in the central part at the top. What you see is his bacteria, and the small circles at the very top on the left are different staph phages. In the middle, you have something called phage bioderm that has mixture, the cocktail, the piophage they use for many things, that's a mixture of staph and strep and pseudomonas and E. coli and proteus, and it does beautifully. But the interesting thing is the lower right-hand corner there, and what that is the antibiotic disc of the antibiotic that he'd been on IV for a full year. and it's still totally sensitive to that. So it's wonderful to have these new kinds of anti-MRSA and so forth drugs. This wasn't MRSA, it was just ordinary staph, but if it can't get into it and can't work, then you're in serious trouble. Phage was used, and he quite quickly recovered. After about 10 days, he went back home from Georgia, where I had taken him. He was from Toronto. And he only a couple of times later had small amounts, very small recurrences, and they went away within a few days. It appears that what happens is that a little bit of phage and a little bit of bacteria was still in the bone, and when he really, really, really pushed hard for a while, he could manage to stimulate both enough to get it to reproduce. But you have story after story after story of this sort of thing where something is cured. He had been told that unless he had his leg amputated, he would be dead within a year, and now he's back functioning very well. And there was a program on 48 Hours that was looking at antibiotic resistance back in 2001 and picked up that story, and a quarter of it was taken in Tbilisi when I could be there, largely about the story. And a lot of people got interested in phage then, but somehow trying to get phage into the pipelines in any way in the Western world is very difficult because it's something that has been used so long that it's hard to get intellectual property. It's something that a box of phage goes for about $10 in Tbilisi. And to get it to a point where where it can actually be approved is not trivial. Why is it that it works, that it can go into the bone and help when other things can't? René Dubos did a series of experiments back in 1943, which were looking at what happens when you inject mice with phage, because people were saying, how could it possibly work? Wouldn't it be cleared out by the immune system? And what he saw was when he just injected the mice intraperitoneally with phage, you saw it quickly getting into the bloodstream, and a little bit of it getting into the brain, far less, and it was cleared out very quickly. But if simultaneously you injected the mice in the brain with the bacteria, in this case Shigella dysenteria, that phage hit, what happened is instead of the bottom line being what you see in the brain, it's the top line. You see over 10 to the 9th per mil in the brain. In other words, those few phage that could get into that protected place, even across the blood-brain barrier, are able to multiply to very high levels. They stayed there. Most of the mice survived, as opposed to most of them dying. And after a few days, when all of the bacteria were killed, then the levels in the brain dropped, and most of them are quickly gone. In terms of, a lot of people ask about safety and early history and so forth and keep calling it a Soviet kind of thing. Well, it was actually discovered in England and in France, not in the Soviet Union, and went later on through a series of things into being used in Eastern Europe and the Soviet Union, in places where you have primarily a public health approach as opposed to a pharmaceutical competitive approach. But just to talk a little bit about it in the West, by the late 1920s, labs and companies in Europe and the US were making a whole bunch of phage products, and with varying degrees of success. And there were companies like Eli Lilly making it without, by the way, proper control, and virtually none of those products were found to actually have antimicrobial activity. But what you found is that a lot of the work that you see coming out from that period of time, much of it very good, is coming from small labs of various sorts. Now, Felix de Herelle himself, you need me to pick this up? Felix de Herelle himself set up a company in France that made phage that were very tightly controlled. In the 1930s, there was a report by the AMA, a very extensive year-long report that talked about most phage not being able to be proven in terms of looking through about 100 papers in English. Of course, they hadn't read any of the French papers. And at about that same time, there was a full issue of La Médecine in France that has some quite incredible descriptions and discussions of all of the data and details of what was happening there. So there are various reasons why we don't see things happening. Why am I saying that staph phage is probably the best for proof of principles using collaborative trials? Well, first of all, the one thing that 1930s AMA study said was that staph for use in various kinds of wounds and ulcers was the one thing for which there was very clearly sufficient data that it was definitely working. It was extensively used in France into the 1990s. Ward McNeil at Columbia in the 1930s and 1940s had hundreds of patients with successful treatment and good data. The Eliava Institute actually made a version of it that was used intravenously back in the 80s to try that. And by the way, it is not in general used intravenously, phage products, anywhere in the world. What they found was that it worked beautifully, for example, this intravenously, this very highly purified preparation on a group of about 200 infants in the hospital in Moscow who had severe septicemia. These were premature infants, so not much in the way of immune systems. Worked in other patients with reduced immune systems, and there were definitely complications in other places. They've taken one of the phages that was in that and highly purified it now, and it was sequenced by a grant I was a co-PI on about a dozen years ago that came from the National Institutes of Health, and it's been developed and purified and so forth into a product that is sold in their stores along with the POphage and the intestiphage. Now, this is a highly purified sequence phage. POphage is for all purulent kinds of infections, and it's a combination of dozens of phage against staph and strep and pseudomonas and E. coli and proteus, and that's what was used in that first treatment that I saw. Intestiphage has phage against 28 different kinds of gastrointestinal problems, but so this phage is one that we're now using. This is, by the way, some Polish results with staph again showing up to be very good. Staph phage lysate has been used in this country primarily, well, it was used both to treat animals and humans, and it was approved for both into the 90s. And in fact, just to put in a little special touch from being here, Elizabeth Taylor, back in 1961, had a very severe staph pneumonia when she was filming Cleopatra, and there's a long story of how it went about that they heard about staph phage lysate being available in New Jersey and had it specially flown over within a couple of days, and that's what she was treated with, and she went from being on a ventilator to clearly recovering. All sorts of stories. But the staph phage lysate has been used so much that there would be at least a great deal of safety data, though in the 1990s it was decided that they didn't have enough other kinds of data. So, and was a compassionate use trial done, which I don't have time to talk more about, using a mixture of phage from Georgia, which included this phage. Just to use a couple of things to show other ways of using it for diabetic ulcers. Randy Fish, who's the one working with us, has been using it on a series of patients. In this case, this was a person who had very poor circulation, had been 5 months on antibiotics with no improvement, the MRI looked suspicious for osteomyelitis, and they started phage, and that's bone showing along the right-hand side of the wound. In this case, as opposed to the first one, he didn't take out any of the bone. He left it there, even though clearly it was probably osteomyelitic. And you can see that within a few weeks, it was largely healing, and by the time you got through, it completely healed up, and there's been no sign a year later of its having come back. This is a case that was very recent, a fellow with necrotic toe, gangrene, and so forth. The first two pictures are taken on the same day after the gangrene was healed away, and here he did cut out the bone. But this person he hadn't treated before. Was sent to him to simply to amputate the toe. That was his assignment. And he decided, though, that the underside of the toe seemed to be quite pink and quite healthy, and went ahead and decided to try to use phage, and again, with very good results. The AstraZeneca drug discovery and development got interested in phage in some ways back in 2001 and had a symposium on drug discovery and development in Bangalore, India, that they invited me to be the keynote speaker for. And the person who wanted to introduce me was Gary Skolnick from Stanford, who was head of infectious disease there then. And he wanted to introduce me to tell about this story, about his mother who had been dying of typhoid fever back in 1948, had been in the hospital with high fever for several weeks, and he had been taken to say goodbye. And his father read about a person in Los Angeles who was working with mice who had phage against typhoid, and he called him and he sent the phage up and injected it, and within 24 hours she suddenly was fever-free and lived for another 40 years. And he said, that's real infectious disease experimentation, a mix of science and daring and desperation. But actually, and he thought that it was something not known, only later we learned there was much data and a solid foundation for this life-saving decision, and a number of hospitals here, one in particular in Los Angeles, were using it regularly, and the description was exactly that, and they had a great drop in what was going wrong. So, and described it exactly the same way. As we explore the possibilities of using phage to help postpone and combat the crisis of antibiotic resistance, it's worth thinking about that challenge. Are there ways we can use diabetic ulcer work to help speed up accessibility to some of the best understood phages while enriching people's lives? And we know a lot about the phage now. So I hope some of you will think about wanting to explore in various ways. And what we'd like to do is to do more of these case studies and get more people involved where it's simply added to a completely normal treatment program, and get a lot of basic data from compassionate use studies to be able to design clinical trials that will be really appropriate for the right uses. And I have 5 vials of those phage along with me, and if people want to talk with Randy, if there are people here who would like to be among those to try that, I'm open for it.
Thank you. Thank you very much. I think we're going to have a short panel if we can have the speakers come up. Well, people are coming up. Saturday, the hands-on workshops are going to be back in the hotel. They begin at 7 a.m.